Hello, my name is Dr. Rick Sims and I'm the Chief Medical Physicist at Auckland Radiation Oncology. I'm going to talk to you today about our clinical experience of ImSimQA, which is the image QA software provided by Oncology Systems Limited in the UK. So this presentation I provided at their booth during the Astro 37 conference in Barcelona earlier this year. And I hope you find this informative and useful. So just a quick overview of the presentation. I'll quickly give you an introduction to the private radiation therapy facility where I work, Auckland Radiation Oncology. I'll then briefly describe how we assisted OSL with importing the deformation vector field from RayStation into their ImSimQA software and how we validated that import. I'll then cover some practical experience of using the ImSimQA software and provide a quick summary and my contact details. Auckland Radiation Oncology was the first private practice in New Zealand, established in 2008, treating our first patients in November of that year, and we're now approaching our milestone 10-year anniversary, which is very exciting. There are now 3 private radiation therapy facilities in New Zealand, shown with the green circles, and 6 public radiation therapy centres, shown with the yellow. We are an Elekta, RayStation and BrainLab reference site, so we use 3 Elekta Agility or Versa HD linear accelerators, all using the Hexapod 6 degree of freedom couch, and the advanced imaging, for example 4D cone beam. We use RayStation treatment planning system and have done so since 2012, and we've recently become a BrainLab ExactTrack reference site, which we installed on one of our new machines earlier this year. And we were actually the first to bring all these 3 vendors into the New Zealand market in 2008, 2012, and more recently with BrainLab in 2018. And needless to say, with this, all these technologies and these software solutions, we're able to deliver a wide range of treatments, both complex and very simple, using a variety of novel software tools. We went clinical with RayStation DIR in 2014, and we commissioned this system using a variety of in-house methods. And all of this was pre-TG132 recommendations and pre-ImSimQA. Now obviously, I'm sure you can imagine this was a significant undertaking by physics, the radiation therapist and the radiation oncologist team, and we used a variety of methods which included physical phantoms and time-consuming contour analysis. A few years later, we acquired ImSimQA for some other projects we were looking at, and we worked very closely with OSL to support the import of deformation vector fields from RayStation into ImSimQA. And needless to say, the tools available to ImSimQA would very much have streamlined this process. Here's just a screenshot covering the recommendations of AAPM TG132 with regards to QA and QC of image registration algorithms. I'm not going to run through all of these individually, but you can see that ImSimQA really does provide all those tools that you need to commission such systems. And this would be far more expedient than using physical phantoms and time-consuming ROI and POI analysis. And so I highly recommend this software for this purpose. Okay, in terms of validating the DIR from RayStation, in terms of its import into ImSimQA, as I said, we worked very closely with OSL to perform this work, and we performed this with RayStation version 4.7. Now, RaySearch are able to provide users with a Python script to export the deformation vector field data from the TPS and into 4 files, which will include a header file and then 3 binary files covering the 3 translation directions. Now we performed a validation of this import into ImSimQA in 3 main steps, covering visualization of deformation vector field, POI analysis, and ROI analysis. And I'll go through each of these in turn with the system. So DIR is commonly used to assess retreatment doses or perform adaptive replanning by transferring the previously delivered dose and the previously contoured structures from the original CT onto the new CT to help guide the clinical judgment. Now in RayStation, we perform this by setting the original CT as your target and the rescan CT as your reference. You follow the appropriate workflow and then you can review the deformation vector fields and the deformed data. Now in RayStation, the DVF is actually displayed on the reference image set, so on the rescan CT in this example, and it shows what DVF needs to be applied to the reference image to deform it into the target. And so for the visualization validation of the RayStation DVF, we need to make sure that in ImSimQA, when we visualize the DVF, it's visualized on the correct data set and in the correct direction, so not in the inverse direction. So I'm going to show an example here on the lower 2 pictures. So on the left is an example of an original CT with the bladder contoured in yellow, and on the lower right we have the rescan CT, and you can see the bladder is in a slightly different position. So once you've gone through the workflow in RayStation, what you would be presented with is the vector field as shown here. So you can see that now RayStation is showing that the post-inf part of the bladder needs to be pushed more posterior to match the original CT on the left. And so it was important during the visualization validation process that ImSimQA also displayed A, the vector fields in the correct direction, and B, on the correct data set. And the results can be shown here. So on the left now is the DVF exported from RayStation, imported into ImSimQA, and overlaid on the correct data set. So that was very good. The second part of the validation process was to do some spot checking on POIs located at various landmarks and throughout the deformation vector field. And then we compared the position of these points, the displacement to the DVF value at these points in the X, Y, and Z directions. The lower left shows the color wash of the DVF on the same data set within RayStation. Then we have the vector field shown in ImSimQA on the inset, and some examples of the numbers on the right here. And what we found actually was that the vector difference between the 2 systems for several different data sets was typically 2.3 plus or minus 0.6 millimeter at a 95% confidence interval. And the DIR resolution used throughout this process was 2.5 millimeters. And so, we were very happy with this result. So it's typically within 1 voxel, which we thought was excellent agreement. The third and final part of our validation of the DVF import was using ROI-based analysis. And this was performed by comparing the original and the deformed contours within each system. So we had the original and deformed contours in RayStation, and we performed contour analysis with in-house scripting. And then we imported the DVF into ImSimQA and performed similar contour analysis using the tools available in that system. And then we compared the results from both systems. And what we found was that the mean distance to agreement and the center of gravity distance difference between the 2 systems was sub-millimeter. So it was excellent agreement. On average, the volume difference was 0, but there was a variation depending on the size of the organ at risk that we were considering. And typically for the bladder in this example, it was within 9 cc, which also represents a sub-millimeter difference in terms of contour position. Then we also looked at the conformity index and the dissimilarity coefficient, and they were all excellent within 0.1 of each other. So in terms of validating the DVF import into ImSimQA, we were very happy at this stage that all the differences we were seeing were sub-millimeter and or at the level of the DIR voxel. Now it's important to create commissioning data to repeat this process when we have new versions of either ImSimQA or the TPS. So I'll now cover some of our practical experience of using ImSimQA. So one of the first examples I'll show you of our practical experience is how you can use the tools in ImSimQA to commission your DIR algorithm. So I'll show an example here for inverse consistency, which is one of the checks suggested by TG132. And this is essentially ensuring that, or at least testing, that regardless of which direction you perform the DIR algorithm process, the results are equivalent. So we performed this test pre-ImSimQA using contour analysis. And so we'd perform the DIR in both directions, produce deformed structure sets, and check the similarity using a dice similarity coefficient. And this image here is showing two different bladder contours shown in yellow and the union of the two shown in brown to allow us to calculate the DSC. And in this example, the results were between 0.97 and 0.99 for a variety of different volumes of bladder. Now, although this seems actually pretty good in terms of the dice similarity coefficient, you can see here that there is a large difference on the anterior superior portion of the bladder, which may or may not be of concern. One of the drawbacks with doing this in-house contour analysis is that we're limited to assessing the DVF differences just along the contour. And so we performed this analysis plus or minus a few centimeters for each contour to produce a bit more spatial information. So whilst this was useful, this allowed us to test for inverse consistency. It's a very time-consuming process and yielded limited information. So this process can be sped up significantly using the tools available within the deform module in ImSimQA. So this system now allows us to import the DVFs from RayStation in both the forward and inverse direction, and to perform some batch processing where the so-called ICE is calculated. And this is a very simple interface and provides very detailed information, which I'm going to show here on this slide. So the bottom left image is showing the contour of the bladder and a color wash showing the difference in the DVFs in the forward and inverse direction. You can hover your mouse over this color wash and it provides you the difference in terms of, or the vector difference in terms of the X, Y, Z position. But more useful is the so-called DVF histogram shown on the right hand side here, which allows us to really drill down and see what the volume of the bladder is within a certain distance. So for example, in this case, we can see that 95% of the volume of the bladder is contained within 0.5 millimeters, and the mean difference is 2.15 millimeters. The maximum difference is 9.3. So this is really useful information, and it's far more detailed than the in-house analysis I've shown previously. If we were to compare the results of both the in-house and the ImSimQA process, they are actually very similar. So the color map on the right hand side at the anterior superior portion of the bladder is showing a difference of between 6 to 7 millimeters, which is consistent with the measurement on the left within RayStation. And in terms of the RayStation algorithm for inverse consistency, we found the largest difference is near regions of air and or external contour. And this is really, well, what's enabled us to really identify this is this additional spatial information in ImSimQA. Another example of using tools within ImSimQA is when you have an upgrade to your TPS, and there may or may not be an upgrade to the algorithm, the deformation algorithm. And so using the deform module, we're able to test for subtle changes in the DVF between TPS versions. And again, this is where having benchmark or baseline commissioning data to rerun these tests over time is very, very useful. Once again, this tool uses the DVF histogram, so you can add or subtract DVF files to see if there's been any difference. And we've previously tested this again using contour analysis and dose mapping. Now, an interesting example of using the deform module with ImSimQA is shown here when we upgraded our TPS from version 4.7 to 6.1. So we've established some baseline commissioning data, and this allows us to just rerun the deformation process in the new version of the TPS, export the DVF to ImSimQA, and compare it with the DVF from version 4.7. Now, the vendor stipulated there's been no major changes in the DIR algorithm between these two versions, but clearly we can see there has been a difference. So we're able to feed this back to the vendor and start a discussion on why these changes were present. In this particular example, we can see that 95% of the voxels for the brainstem are within 0.2 millimeters. So we're not talking particularly large differences. The DVF resolution is 2.5 millimeters, but nevertheless, it's a really useful tool to identify these changes, which may or may not be larger than this. The second area of our practical experience I'll cover is the contour analysis area, or the contour module in ImSimQA. And this is an incredibly powerful module that provides many different options for comparing contours on a CT image set. There are various volume and distance-based metrics that are produced slice by slice or for the entire organ at risk. And also we're able to visualize the results, look at a pass or fail using contour distance maps, and also export text-based files of the data. Now, we use the contour comparison modules extensively for various research and development projects, and also routine assessment of inter-user variation within the planning team. The process by which you perform contour analysis is incredibly simple. You select the relevant CT data, you select multiple RT structural files, and then you can choose to have automatic processing or perform this manually. This is just showing an example of the contour analysis module for a breast patient that formed part of a poster that we presented at Estro in Barcelona. This was a study where we were comparing the inter-user variation in contouring a novel implantable marker, and also then comparing the resulting CTVs and PTVs. And so you can show here on the left, you can see you define your evaluation contour and your reference contour. You merge these contours, clicking the merge RT structures, or by using the batch processing. It performs the entire analysis and then produces this table shown at the bottom. And you can see all the different metrics. You have conformity index, mean distance to agreement, various volume-based metrics, and for example, dissimilarity coefficient. So it's incredibly powerful for assessing these contours very, very quickly. Shown on this slide is the detailed comparison results for a particular contour pair, in this example CTV, and this really allows you to drill down on the slice by slice level to look at the agreement either in a volume or distance based metric between the two contours. It also allows you to look at the volume of mismatch between the two contours in terms of under contouring or over contouring. So it's very, very useful to drill down into that level of detail. The detailed slice by slice differences between your evaluation and reference contour can be seen visually using the contour difference map. An example is shown here for the biorb contour, where the reference and the evaluation contours are within a few millimeters of each other, and this has been defined as acceptable and so is shaded in green. You can also define an area where the agreement is not acceptable, and in this example it's defined as over 5 millimeters, and here it's shaded as red. So this is a very useful, very visual based comparison tool to assess contour differences. This is just shown another example of using the contour analysis tool for a prostate patient, and this is a collaboration we're performing with Microsoft in terms of machine learning algorithms. Now, the real beauty with using this tool is that it allows you to analyze this data very quickly. So for this study we have 200 to 300 patients that we're analyzing, which can be up to around 1,000 contours. And so doing this manually would be incredibly time consuming. And so it's very powerful for this kind of application. The third and final example of our practical experience I'd like to show you is our use of the Jacobian determinant calculation in the deform module in ImSimQA. Now this is incredibly useful for investigating areas of problems or problem areas within your DVF by visualizing areas of contraction, expansion or folding in the DVF, which aren't always obvious when looking at the vector fields in RayStation, for example. Now, the reason this is incredibly useful for us is that in RayStation, when you visually look at your DVF, it's not always obvious that there are areas of folding. And so what happens when you try to invert this DVF to convert or transfer your doses and structures onto your different data set is that it fails. It gives the error message that it's non-invertible. And so this allows us to really drill down to identify where it's being folded and to hopefully optimize our deformation process for these cases. Now this is an example of a problem case in RayStation where the DVF was non-invertible. And so we've imported the CT images and the DVF into ImSimQA, and we've visualized the DVF as shown here. Now it's not immediately obvious where the problem areas lie, and so that's why calculating the Jacobian determinant is so useful. And here we have the results of the Jacobian determinant on that data set. And we can see areas of expansion, which are shown in blue, of contractions shown in yellow, and most importantly the red showing the folding. Now it's these folded voxels that cause issues with RayStation when it attempts to invert the DVF to allow you to transfer the RT dose and structure files. What's very nice with this software is not only does it visually show you where it is, it also provides some statistics to show you the percentage of folded voxels both within the DVF and within the body. And this now allows us to go back to RayStation to optimize the DIR process to hopefully remove areas prone to folding. So it's been very, very useful for that purpose. In summary, ImSimQA provides extensive tools for commissioning both rigid and deformable image registration algorithms, and it's very much aligned with the recommendations from TG132. The tools available within the software are invaluable for routine TPS upgrade checks, for investigating any issues with DVFs from your TPS, and for performing contour comparison studies. Now ImSimQA also has a host of other features which I haven't touched upon, for example creation of virtual phantoms, and OSL will be able to help you with any questions you may have. So I hope you've enjoyed the presentation. I hope it's been useful. I'm happy to receive any questions on my email address shown on the slide, and thank you for listening.